Hi, my name is Cheng-Yu Lee. Today I'm going to talk about the role of prostaglandin F2 alpha in female reproductive signaling in an African cichlid. The broad interest that I have is to understanding how does an individual perceive and process social information and then translate into adaptive behavior changes. And in this current project, I use Astatotilapia burtoni cichlid as a study species and then focus on olfactory cues. And we ask, can males perceive the differences between gravid and non-gravid female solely by odor cues? And if yes, what are the key chemical cues driving this preference? And what are the receptors detecting these cues? So to answer this question, we built this preference test setup in a 30-gallon tank. And there are two odor tanks connecting to parasitic pump, which deliver odorant into this preference tank. And this odorant can diffuse through this permeable divider into each preference zone. And then we recorded the 90 minutes preference behavior before and after delivering odorant into the tank. And we use 3 different ways to calculate the preference score. And due to the time limitation today, I only present the percentage of time that males spend in each preference zone as a preference index. And the first question is, can males perceive the differences between gravid and non-gravid females solely by odor cues? And the answer is yes. So in this bar graph, the yellow and blue portion represent the time that males spend in each associated zone. And before we deliver odorant into the tank, you can see male did not show any differences between the time that's spent in each zone. And after we deliver female cue into the tank, the males prefer spend more time in the zone associated with gravid female cues. And we also did a similar test in adult female and in juveniles, and they did not show any preference for gravid females, suggesting that this is a male-specific preference for gravid female. Then what's the key component in the olfactory cues for driving this male's preference? So we hypothesized that prostaglandin F2 alpha, PGF2 alpha, should be a potential female pheromone because in most epithelial species, the level of PGF increases when close to spawning. And in zebrafish, PGF is also one of the chemical cues to attract males. And in some other cypriniform species, the male is also sensitive to PGF metabolite. However, whether cichlid use PGF as a female pheromone signaling is still unknown. So to test this hypothesis, we use indomethacin to block the prostaglandin synthesis pathway. So in this experiment, the first part is to test the male's preference between gravid female and non-gravid female. And we know that the male should show strong preference for gravid female, just like what I show you in the first experiment. Then we inject both female with indomethacin, and this should significantly reduce the prostaglandin levels, particularly in this gravid female. Then we test the preference again to see if male still shows strong preference for this gravid female after inject with indomethacin. And what we found is that before inject with indomethacin, the gravid female has significantly higher PGF compared to non-gravid female. And the males show strong preference for this gravid female. However, after inject with indomethacin, there is a significant decrease in the PGF level in gravid female. And the male is no longer showing any preference for this gravid female after inject with indomethacin. So this result indicates that indomethacin abolishes gravid female attractiveness to the males and indicating the necessity of prostaglandin in mediating female reproductive signaling. And we further discovered that the males are insensitive to PGF2 alpha or 3 major metabolites. And males are also insensitive or show no preference in our preference test for these 4 chemicals. But males are sensitive and show strong preference for non-gravid female, which are injected with PGF2 alpha. And this strong preference cannot be found in a female injected with PGE2 or PGD2. And this preference can be independent of PGF hormonal receptors because if you inject PGF2 alpha into the knockout female, they are still attracted to the males. So our current explanation is that in cichlid, PGF is not a pheromone to directly induce male's preference, but it drives reproductive female pheromone signaling through its metabolization. Therefore, PGF is converted to PGF metabolite and driving these male's preference. Then what's the receptor detect gravid female cue and PGF metabolite? So fish use olfactory epithelium to detect odorant in water. So this odorant can bind into a receptor which expressed in these olfactory sensory neurons, or we call it OSN. And there are two major categories of OSN. There are microvirus OSN and ciliated OSN. And in this experiment, we ask which population of OSN are responsible for detecting these gravid female cues. So to answer this question, we expose males to gravid PGF-injected female, non-gravid female cues and use tank water as a negative control. And we use CFOS RNA probe to label those OSN which are activated by these female cues. And we also use TRPC2 probe to label microvirus OSN. And we found that gravid female activate more OSN compared to non-gravid female or tank water exposed group. And the majority of these activate OSN are ciliated OSN. And we further discover a subset of olfactory sensory neurons which express beta group olfactory receptor. And these beta OR OSN are only activated when animal exposed to gravid female and PGF-injected female cues. So suggesting its role for detecting the fertility status of females in burtoni cichlid. So in conclusion, we know that the male cichlid can differentiate gravid versus non-gravid females solely by odor cues. And the PGF is necessary and sufficient to induce this preference. And though it did not serve as a pheromone cue to induce male's preference, but it acts as an indirect pheromone to mediate female attractiveness to conspecific males. And it's possible go through this PGF metabolite pathway, and this PGF metabolite activates male's OSN to induce male's preference. And then lastly, we found that a subset OSN expressed beta OR could be responsible for detecting these gravid female cues. So thanks for your attention. I'm sorry I won't be able to be there to answer your question today, but my advisor, Dr. John T, will be there and he's happy to answer any questions. Thanks for your time.